Hello everyone, welcome back to Gray's Gyan, your go-to channel for medical anatomy made simple. Today we're diving into one of the most elegant and underappreciated structures in the entire human body, microvilli. So what exactly are microvilli? Imagine you're painting a wall, and you look at your paintbrush. You see all those tiny bristles pointing outward. That's exactly what microvilli look like on the surface of a cell. Hundreds and thousands of tiny fingers, all pointing outward. Now let's get precise, because this is Gray's Anatomy, and we love precision. Each microvillus is roughly 0.1 micrometers in diameter. That's incredibly thin, and up to 2 micrometers long. They are covered on the outside by the plasma membrane, and on the inside they have a core of actin filaments, which we'll talk about in just a moment. When you look at them under a light microscope, and they're all lined up neatly in a row, they look like the bristles of a paintbrush, and that's exactly why they're called a brush border, or a striated border in histology. Now, where do we find these structures in the body? The two most classic locations are: Number 1, the small intestine. Specifically on the absorptive epithelial cells called enterocytes. After you eat food and it gets digested, your intestine needs to absorb all those nutrients as efficiently as possible. Microvilli dramatically increase the surface area available for absorption. That's their primary job here. Number 2, the kidney, specifically in the cells lining the proximal convoluted tubule of the nephron. Here, microvilli help reabsorb glucose, amino acids, ions, and water from the filtrate back into the bloodstream. Again, maximum surface area for maximum reabsorption. Both of these locations form what we classically call the brush border, a beautiful, dense carpet of microvilli when viewed under the microscope. Now let's open up a single microvillus and look at what's inside. This is where it gets really interesting. Think of it like this: imagine a tent pole wrapped in a tarpaulin. The pole is the structural support, and the tarpaulin is the covering. In a microvillus, the actin filaments are the tent pole, and the plasma membrane is the tarpaulin. Inside each microvillus, you have a tightly packed bundle of 20 to 30 actin filaments running from the base all the way to the tip. These are held together by cross bridges made of two proteins, fascin and fibrin. Think of these as tiny clamps keeping the actin rods bundled neatly together. Now, these actin filaments also need to be connected to the surrounding plasma membrane, otherwise the whole structure would just collapse inward. That connection is made by bridges of myosin 1 and calmodulin. They act like guy ropes, linking the inner actin bundle to the outer membrane. And at the very tip of each microvillus, at the top, the free ends of the actin filaments are inserted into a dense protein mass that contains a protein called vilin. Vilin is essential for maintaining the integrity of the bundle at the tip. This entire structural model was beautifully described by Brown and McKnight in 2010 in their landmark paper published in PLOS 1, titled Molecular Model of the Microvillar Cytoskeleton and Organization of the Brush Border, from Boston University School of Medicine. Absolutely worth reading if you want to go deeper. So the actin bundle runs all the way through the microvillus. But what happens at the base? Where does it go? It plunges downward into the apical cytoplasm and gets anchored into a structure called the terminal web. The terminal web is like the foundation of a building. It keeps all the microvilli stable and correctly positioned. It consists of transverse actin filaments that run perpendicular to the microvilli, stabilized by a protein called spectrin. Underneath all of this, there are keratin intermediate filaments that provide additional support. The web is also anchored laterally to the cell's junctional complexes, specifically the tight junctions and the zonula adherens, at the apical borders of the epithelial cell. And here's something fascinating. The terminal web also contains myosin 2 and tropomyosin. These are contractile proteins. This tells us the terminal web isn't just a passive scaffold, it likely has contractile activity, meaning it can actively regulate the arrangement of microvilli. Exactly how and why is still being studied. Now let's talk about something special that sits on top of the microvilli in the small intestine, the glycocalyx. If you look at the electron micrograph here, this is figure 1.27 from Gray's Anatomy, courtesy of Dr. Bart Wegner from Sheffield Teaching Hospitals. You can see that fuzzy, hazy coat sitting at the tips of and between the microvilli. That fuzzy coat is the glycocalyx. So what is it? It is formed by the extracellular domains of integral membrane glycoproteins, essentially, sugar-coated proteins whose outer portions project out from the membrane's surface like antennae, creating a thick, sticky coat. And this coat is not just decoration. It is loaded with digestive enzymes, things like lactase, sucrase, and aminopeptidases, responsible for the very final steps of digestion. So your digestion is literally happening right on the surface of these microvilli. Clinically, this is very important. In conditions like celiac disease or rotavirus infection, this glycocalyx gets damaged, and the result is enzyme loss, malabsorption, and diarrhea. Let's now appreciate just how powerful microvilli are when it comes to increasing surface area. Microvilli increase the surface area of the cell by up to 40 times. 40 times. Here's an analogy. Take a tennis ball and flatten it. You get a small circle of area. Now, imagine covering that entire circle with thousands of tiny fingers. Suddenly your absorptive area explodes. The small intestine, without microvilli, would have a surface area of roughly 0.5 square meters. With microvilli, taking into account the villi, plicae-circulars, and microvilli, the total absorptive surface area reaches approximately 200 square meters. That is roughly the size of a tennis court. Inside your abdomen. That is the engineering brilliance of microvilli. Now, not all microvilli are neatly arranged in a brushed border. Some are irregular and variable in shape, and these are called filopodia. The word comes from Latin phylum meaning thread, and Greek pos meaning foot, so literally, thread feet. Filopodia are found on the surfaces of cells like free macrophages and fibroblasts. Unlike the regular, absorption-focused microvilli, filopodia are associated with two very different functions: phagocytosis, where the cell engulfs particles or pathogens, and cell motility, where the cell uses these projections to crawl and move through tissues. So the same basic structure, a membrane-covered actin projection, can be tuned for very different purposes depending on where it is and what the cell needs. Now we come to one of my favorite topics, and it comes with a naming controversy. Stereocilia, or as they should more accurately be called, stereovilli, are long, branching microvilli. Here's the important point: they are not cilia. This was a historical mistake in naming that stuck in the literature. True cilia have an internal skeleton made of microtubules arranged in a 9+2 pattern, and they can move. Stereovilli have actin filaments, just like regular microvilli, and they are completely non-modal. The name, stereocilia, was given before this distinction was properly understood, so the correct term is stereovilli, and Gray's Anatomy uses both terms but acknowledges the misnomer. Where are they found? Firstly, in the cochlear and vestibular receptor cells, the hair cells of the inner ear, where they act as sensory transducers. They convert mechanical movement into electrical signals. Secondly, in the absorptive epithelium of the epididymis, the coiled tube where sperm mature, where they help absorb excess fluid from the lumen. Let me walk you through how stereovilli help you here, because it's genuinely beautiful biology. Step 1. Sound waves enter the cochlea of the inner ear. Step 2. These waves cause the endolymph fluid inside the cochlea to move, which bends the bundles of stereovilli on the hair cells. Step 3. When the stereovilli bend toward the tallest ones in the bundle, tiny protein links called tip links get stretched, and this mechanically opens ion channels at the tips. Step 4. Potassium ions rush into the hair cell, causing it to depolarize. Step 5. This depolarization triggers the release of glutamate as a neurotransmitter, which excites the auditory nerve fibers, which send the signal to your brain, and your brain interprets it as sound. In the vestibular system, responsible for balance, the same mechanism works but detects head movements and gravity. Bending in the opposite direction closes the channels, causing hyperpolarization. One important note: in mammals, damaged cochlear hair cells cannot regenerate, so protect your hearing. Before we wrap up, let's give credit where credit is due. The landmark paper that gave us our best molecular model of the microvilli cytoskeleton is Brown J.W. and McKnight C.J., 2010. Molecular Model of the Microvilli Cytoskeleton and Organization of the Brushed Border. Published in PLOS 1, Volume 5, Issue 2, Article E9406. This work came from the Department of Physiology and Biophysics at Boston University School of Medicine, funded by an NIH grant and a Boston University Graduate Student Research Fellowship. Their key contributions: they built the first complete in silico molecular model of the brushed border, clarified how each protein fits into the architecture, showed how approximately 19 actin filaments are cross-linked to span the 100 nanometer diameter of a microvillus, and established the hexagonal arrangement of microvilli in the brushed border. And the beautiful electron micrograph we've been using throughout this video, Figure 1.27, is courtesy of Dr. Bart Wagner from the Histopathology Department at Sheffield Teaching Hospitals NHS Trust in the UK. Thank you, Dr. Wagner. Alright, let's do a rapid revision before we go. Microvilli, finger-like projections, 0.1 micrometers wide, up to 2 micrometers long. Core, 20 to 30 actin filaments, cross-linked by facin and fibrin. Membrane connection, myosin 1 and calmodulin bridges. Tip, vilin protein at the free barbed ends of actin. Base, anchored in the terminal web made of spectrin, keratin, myosin 2 and tropomyosin. Function, 40 times surface area increase, site of glycocalyx with digestive enzymes. Filopodia, irregular microvilli on macrophages and fibroblasts, phagocytosis and motility. Stereovilli, long and branching, no microtubules, found in the inner ear and epididymis. And that's a wrap on microvilli. If you found this video helpful, please give it a thumbs up, share with friends, and if you're new here, subscribe to Gray's Guion for more informative videos like this.